Hello everyone, BiyokimyaVET channel is here with the topic titled Lipids-I. Do not forget to like the video before you start watching it, my friends. I wish it to be useful. Let's start. The contents of the topic are Introduction to Lipids, General Functions of Lipids, Classification of Lipids, and the major title is today Fatty Acids. We will study saturated and unsaturated fatty acids, the nomenclature of fatty acids, classification, essential ones, and at the end, physical and chemical properties of fatty acids. Let's start with introduction to lipids. Lipids, together with carbohydrates and proteins, are a group of organic substances found in nature and in organisms and are of the greatest quantitative importance. They can be used by all living organisms, and they build up some critical parts of organisms, such as cell membranes. Cell membranes are lipids, or they have lipid character, or any membranous structure in the organism carry lipids. Lipids also are functional components. Some lipids provide integrity to the organism, or provide energy to organism, or provide some functionalities similar to the hormones. They are organic molecules that are generally insoluble in water and soluble in solvents such as ether and chloroform. Their most important properties are they are a source of energy. They can give more energy than carbohydrate and proteins, but not all lipids. This is only eligible for fatty acids. It must be present in the structure of membranes. So this is the second major significance of lipids. They are esters of fatty acids or can esterify. So this is very basic definition. As in the carbohydrates, it's not to put an exact definition covering all lipids, so we approach more general, and this is the one. They are esters of fatty acids or can esterify. Lipids have a unique place among organic molecules. The identity of lipids is based on their physical properties, not on the presence of specific functional groups they contain. Therefore, they have very large and different structures, and each one has a different role to play. Lipids contain a very high number of nonpolar carbon-carbon single bonds and carbon-hydrogen bonds. As a result, as a basic definition, lipids are nonpolar or weakly polar biomolecules, soluble in organic solvents such as chloroform, carbon tetrachloride, but insoluble in polar solvents such as water. And as you see, lipid carries a nonpolar structure because lipids have many structural features in common with hydrocarbons. In addition, most lipid molecules contain several polar bonds. They carry a carbonyl group, which makes them polar at the site of carbonyl group. And so these polar structure provide them interaction with various other functional groups. Yes, general functions of lipids that I have already told about two of them. Which one is? They are cell membrane components. And the cell membrane surrounds the cell, protecting it from external influences and enabling metabolic activity within the cell. Membranes are not a simple structure surrounding the cell, but contain many important enzymes and transport system. As you see here, this is a very complex, highly organized structure which the some lipids in the cell membrane structure, as you see here, the green headed ones and two tails, which are the phospholipids, presentation of phospholipids, which is commonly accepted representation style. They provide the main structure of the cell membrane, but also cell membrane contain carbohydrate moieties attached to proteins or lipids. Cell membrane contain proteins, whether they may be peripheral protein or integral protein, and also cell membrane carry cholesterol as a building component. So the membranes, cell membranes, or any membranous structure contain many specialized structure, and in the cell membrane they carry receptors, transporter proteins, which they interact with the hormones or other substances. Also, they have function in cell-to-cell communication. They provide a kind of recognition size that cells knows what's happening surrounding cell or in the neighboring cell. And here is a more zoomed look at the lipid layer structure. As you see, the polar head, a kind of pin head shape, and the two tail, which are the non-polar, and this is the polar. And here, as you see, two tail represent the two fatty acids here in phospholipids, or assume if it's a sphingolipid, it represents one fatty acid and one sphingosine alcohol, and the polar head is a phosphate group, which carry another polar groups, and in general they are amino alcohols that we will look later. And the polar head is an essential part because outside of the cell and inside of the cell, which there is a cytoplasm, the fluid part of the cell, a gel-like fluid part of the cell, cell membrane must interact with the water, so they must carry hydrophilic regions, which the polar head provided. Instead, the hydrophobic tail is located inside of the cell membrane, inside of the bilayer structure, which they are hydrophobic parts. They are hydrophobic. And as the second major function, they are storehouse of energy. But not all lipids provide energy, predominantly triglycerides. And here is a structure of a triglyceride, which carries 3 moles of fatty acid, 3 moles of fatty acids, and also a 3-carbon backbone, which is glycerol. And here you see the molecule stearin, a basic triglyceride carrying 3 moles of stearic acid and 1 mole of glycerin. And they are stored in adipose tissue. And we call them fats in animals, or in vegetable forms are oil, fats and oils. And they are good energy sources, because each triglyceride, as you see here, carries 3 moles of fatty acid, and these fatty acids can be used as an energy source, can be used as a fuel. And the oxidation of fatty acids gives 9 kilocalories per grams of energy. Whether in carbohydrates and proteins, they give 4 kilocalories per grams of energy. So fatty acids gives more energy. What about the other functions? Lipids are the source of some important substances in the organism, such as lipoproteins, bile acids, fat-soluble vitamins, the vitamin A, the vitamin of vision, vitamin D, which is important in calcium regulation, vitamin E, the antioxidant vitamin, and the vitamin of coagulation, vitamin K. And also they are the precursors of steroid hormones, which are so important in regulating metabolism and reproductive reactions or reproductive processes. Lipids are effective in protecting against infections, and here the famous family eicosanoids, which the prostaglandins, thromboxanes, and leukotrienes are the most significant members of this family. And you may remember from organic chemistry, the eico represents 20. So they are the molecules carrying 20 carbon atoms in their structure. And so these are the derivative molecules, a kind of derivative molecules, but also they are classified as lipids. So they provide functionality to the organism, to the cells. Lipids are involved in cellular stimulation, cell signaling. They are lipid messengers. They bind to protein structure target receptors or enzymes, such as kinase, phosphatase, and allows the formation of a specific response in the cell. And there are too many lipid messengers, such as sphingolipid messengers, phosphatidylinositol messengers. There are G protein coupled receptor activators such as prostaglandins and also nuclear receptor activators. The good example is the steroid hormones. It's time for classification of lipids, and there are different styles or different approaches. The watch one is we can classify lipids. They can hydrolyzable with water, and these are the lipids that can be broken down into smaller molecules when hydrolyzed with water. This means water break the ester bonds. Triglycerides, phospholipids, waxes, and sphingolipids are the members. And non-hydrolyzable lipids, which they cannot be broken down into smaller molecules or their building blocks when hydrolyzed with water. And the structure of such lipids is more diverse. Fat-soluble vitamins, steroids, and eicosanoids are the members. So this is one type. And also we can classify them whether they are stored or to be a part of membranes. The storage lipids, the neutral lipids also, they don't carry charge, which are the triglycerides. And membrane lipids, we have the family, maybe the most notable family, the phospholipids. They can be classified into two: glycerophospholipids, the lipids carrying glycerin and is a backbone, and sphingospholipids, which they carry sphingosine as a backbone. And also there are glycolipids, the lipids carrying, glycol represents the carbohydrate. They carry carbohydrate moiety. We can also classify them whether they are saponifiable or the ones that we can produce sapons or non-saponifiable lipids. Also, we can classify them according to their structure: simple structure lipids, derivative lipids, and the more complex structure lipids. And the classification which I follow in my presentation. First, we will study fatty acids, which we can accept them as a building blocks of many lipids. The glycerin carrying lipids. The lipids carry glycerol or glycerin as a backbone, which there are two classes, the neutral lipids, and when we will study triglycerides, then the phosphoglyceride, the phosphatase, the phosphatidylglycerol, and phosphatidylinositol. And then lipids not carrying glycerin, which they carry different building blocks or different patterns, the sphingolipids. And under sphingolipids, we have ceramides, sphingomyelins, and glycosphingolipids, which the sphingomyelins are the sphingospholipids. So in the previous slides, I talked about the phospholipids, which there are two classes, glycerophospholipids and sphingospholipids. In this classification, one is in the glycerin carrying lipids, the another one is under lipids not carrying glycerin. Then waxes, terpenes, and sterols. And at the end, we will look to lipids linked to other classes of compounds or more complex structures, which we will mainly study: lipoproteins and the eicosanoids. And so this is the classification. It's time to study fatty acids. We will study first the definition or a basic introduction to fatty acids, then definition of saturated and unsaturated fatty acids, the nomenclature, classification, and the other titles. Fatty acids, shortly Fa, constitute the most important class of lipids and are long-chain organic acids, which means they carry carboxyl group with from 4 to 28 carbon atoms. Fatty acids contain a hydrocarbon tail and a carboxyl group. In other words, fatty acids are monocarboxylic. Mono means they carry only one carboxyl group. They are monocarboxylic organic acids with hydrocarbon chains. So they carry hydrocarbon chains, either alkane or alkene hydrocarbon chain. They are mainly obtained from the hydrolysis of triglycerides. In other words, fatty acids are not found free in cells and tissues, and they are insoluble in water and have a fatty character. Most fatty acids form complexes with other lipid molecules, as in the triglycerides or in phospholipids. As I previously said in the previous topic, here again I also want to emphasize fatty acids are not found freely in cells and tissues. And they are not a desirable things because they are acids, so they can affect the pH of the both intracellular and also extracellular environment. But also during the metabolism, or depends on the metabolic rate, there may be free fatty acids, especially in the plasma during their metabolism, but in a very small concentrations. And these are called unesterified, shortly free fatty acids, FFA, or the more common one is non-esterified fatty acids, shortly NEFA. And this form is transported in the plasma with the link to the albumin, which is the carrier protein. And they are removed from tissues, or they size enzymatically or by chemically hydrolysis. Fatty acids are differentiated from each other according to the number of carbons in their structure, or the double bond they carry, and thus chain lengths they contain, which is determined by the number of carbon atoms. Almost all fatty acids in nature have double carbon atoms. Those with 16 and 18 carbon atoms are the majority. And about 100 different fatty acids have been identified from various plants, animals, and microbes. And the majority of fatty acids found in animals are unbranched, straight-chain derivatives, and have an even number of carbon atoms. And here is the famous one, the common one, palmitic acid. And look at the structure here. This is the formula. They carry 16 carbon atom. And look at here, they carry a hydrocarbon chain, and which is an alkane chain. There is no any carbon-carbon double bond. And this is the carboxylic group, which provide a polar structure, the hydrophilic head and the non-hydrophilic or hydrophobic hydrocarbon chain. And this is the palmitic acid. And also it has a UPAC name that we will talk about the UPAC naming in nomenclature title. There are two classes of fatty acids, depends on the hydrocarbon tail they contain. The one is the saturated fatty acids, shortly we can say SFA. They are the fatty acids that have a long hydrocarbon tail and contain only carbon-carbon single bonds, such as palmitic acid, as in the previous slide, and the stearic acid is a 18 carbon carrying fatty acid. And this group includes short-chain volatile fatty acids, VFAs, derived from microbial carbohydrate fermentation, especially occurs in ruminants, and acetic acid is the simplest fatty acid as well as the simplest volatile fatty acid. Only 2 carbon atoms carried in the structure. And the others one are the propionic acid, butyric acid, and valeric acid, or in general, till the fatty acids having 10 carbon atom, they are volatile in room temperature. In animals, palmitic acid is obtained by lipogenesis. This is the most critical fatty acids. And what I said, animals have generally 16 to 18 carbon atom fatty acids, as well as there are 22 ones. But this is the more common ones, or the ones having more concentration or more content in the body. And the smaller ones, generally occurs during metabolism, during synthesis or breakdown of fatty acids as an intermediate metabolites, Unsaturated fatty acids, UFAs. UFAs contain one or more carbon-carbon double bonds in their structure. So their hydrocarbon chain is an alkene chain. But in SFA or saturated ones, they carry an alkane. This one, alkene. If there is only one double bond, we call them monosaturated fatty acids. They only one carbon-carbon bond. If there are more than one double bond, such as 2 or more, 3, maybe 5, they are called polyunsaturated fatty acids. The famous ones, the PUFAs, they're notable, they're significant, because in general the PUFAs are accepted as an essential. So the animals, the mammals, must take PUFAs from food sources. And also, remember from organic chemistry, the stereochemistry, the carbon-carbon double bond maybe have isomers. We call them geometric isomers, cis or trans configuration, due to diversity of the placement of double bonds. And unsaturated fatty acids generally dominate over saturated fatty acids, especially in higher plants and animals living in cold environments. Look at this given fatty acids. As you see, there are 3 fatty acids, which 2 are in cis configuration, cis oleate or oleic acid, and cis arachidonate or arachidonic acid. And so they are, due to they donate protons in the liquid environments, we call them arachidonate, but we can omit them. Thus, we can also say arachidonic acid, allylic acid, or the oleic acid. As you see in the oleic acid, the double bond is in the cis configuration and is twisted where the double bond is appearing, and we have an L-like shape because the substituents are on the same side. This is why we call them cis. What about in allylic acid? Is in the trans configuration and retains its straight form where the double bond is. So they look like a saturated fatty acid, a straight chain, because the constituents are in the opposite sides. Thus we call them trans configuration. Most natural long-chain unsaturated fatty acids have a cis configuration. This is the common ones. And here the arachidonic acid, look at the arachidonic acid, a 20 carbon carrying and a 4 double bond carrying, 4 double bond in cis configuration, and has a U shape. This means the cis configuration here provide fatty acid to occupy less space, and this decreases their melting point. This affects their physical properties, especially melting point. The trans ones are found in small amounts in nature. They are mostly produced in the food industry, such as margarine. The most common one, especially in hydrogenated vegetable oil, is L-idic acid. In addition, L-idic acid is naturally found in ruminants due to rumen flora, but lesser content. Anti-trans fats can lead to some metabolic problems such as hypercholesterolemia, atherosclerosis, and coronary atherosclerosis, and can cause problems in the metabolism of essential fatty acids. But that's also we talk about the cell membranes, that they must carry phospholipids, and at least one fatty acid in the phospholipid structure is unsaturated fatty acid. And it should be in cis configuration to provide the fluid character of cell membranes. The most abundant fatty acids in mammalian living beings are oleic acid, palmitic acid, and stearic acid. And here my friends, did you see there is numbers in parentheses? There are numberings of fatty acids. They are the identity numbers of fatty acids. And in nomenclature, firstly we write the number of carbon atoms, then using colon, and after colon we put the number of carbon double bonds. In oleic acid, which is an unsaturated, there is only one double bond, carbon-carbon double bond. But in palmitic acid and stearic acid, there is no carbon-carbon double bond. Thus, they are saturated ones. But oleic acid is unsaturated. And please be careful, the punctuations are so important. They must be correct. It must be colon here, not the semicolon or not a comma. And here's two famous one or two of these, the stearic acid, 18, a saturated fatty acid, no double bonds, and a long chain structure. But what about an oleic acid, as you see, which is the most common fatty acid, not only most common saturated, or not only unsaturated, most common unsaturated. It is most common fatty acid in general. And it's an unsaturated fatty acid carrying one cis double bond, which there is a band, there is a twisting here in the point of cis double bond, which gives the oleic acid a L-like shape. The most abundant unsaturated fatty acids in animal lipids are palmitoleic, oleic, linoleic, and arachidoleic acid. So these are the most abundant unsaturated ones, the UFAs. And some are monounsaturated, as in the oleic acid, but some are PUFAs, polyunsaturated fatty acids, linoleic acid or arachidonic acid. Oleic acid is the most common fatty acid in nature and has been found in all known natural oils and phospholipids. Mammals are able to synthesize saturated and mono double bonded unsaturated fatty acids or monounsaturated fatty acids. And here the situation can be variable, especially if the PUFAs are accepted as an essential fatty acids that we will look later again. It's time to study nomenclature of fatty acids. Each fatty acid has a generic name according to trivial naming, or they have the common names. For example, oleic acid, stearic acid, palmitic acid, acetic acid, butyric acid, arachidonic acid, etc. But also they have UPAC names. Do you remember the UPAC naming in the previous courses, organic chemistry? You should remember. According to the UPAC, which is International Union of Pure and Applied Chemistry, systematic naming, fatty acids are named according to hydrocarbons having the same number of carbon atoms. In the system, the E suffix of the hydrocarbon are replaced with the suffix oic acid. So if it is an alkane, for example, alkane, as in the hydrocarbon, the E drops and oic acid come, this will be anoic acid. Anoic acid. Or in the alkenes, which the suffix is E, here the E drops and anoic acid come here, it will be anoic acid. Anoic acid, oic acid, sorry, oic acid comes here, it will be anoic acid. And here you see, the saturated fatty acids terminates with the suffix anoic acid. If we have a saturated fatty acid carrying 10 carbons, so remember the U-pack multiplier of 10, this is the deca. If it is hydrocarbon decane, and here the E drops and it will be decanoic acid, and its common name is capric acid. So if we have a 18 carbon atoms in a fatty acid, and it's a saturated fatty acids, so what is the U-pack multiplier of 18? It is octadecane, but this is the hydrocarbon name, and according to nomenclature, what I said, E drops, and the oic acid comes here, so it will be octadecanoic acid, which is the stearic acid. Or in a palmitic acid, which there are 16 carbon, the U-pack name is hexadecanoic acid. So this means, my dear friends, you should remember the U-pack multipliers in naming hydrocarbons, or in naming the carbon backbone of an organic molecule. Give me a second to erase here. Yes. So, if there is an unsaturated fatty acid, it will terminate with the anoic acid. As I said, remember the alkane naming, with the suffix E, so the E drops here, and we put the oic acid. Thus it will be anoic acid. So if there is an 18 carbons and one single bond between 9 and 10 carbons, the name will be 9-octadeca-anoic acid, which is the oleic acid. And here you see, if there is a double bond, according to nomenclature of alkenes also, we have to designate the position of double bond, or in more general, position of functional group. This is the double bond. What about the carboxyl group? Also it is a functional group, but it's the superior group. And according to the nature of the carboxylic group, they are always at the first carbon, and we can omit them in using in the name, because it's a carboxylic acid, it's always the first carbon. There's no problem. It's not essential to determine the monoic acid or monocarboxylic acid, not necessary. And also there is no second or third carboxylic acid group. There's only one carboxylic. They are the monocarboxylic hydrocarbons. And if there is 18 carbons with 3 double bonds, and double bonds in the position between 9-10, 12-13, and 15-16, the name will be 9-12-15 octadeca. And look at here, we have an additional prefix just in front of the En. Each En represents there are double bonds, and 3 means there are 3 double bonds. So these numbers and the number of double bonds here given, or the number of positions here, must be compatible with this prefix. If you give a 3 position, there must be 3 suffix, 3 not suffix, 3 prefix, just in front of the En. Octadeca-3-anoic acid, and the common name is alpha-linolenic acid. And there is one more missing point here, which I omit. If there is a double bond, they are in cis or trans configuration. We also have to determine the configuration of double bonds. And what I said, in nature, general cis configuration. And sometimes we omit to put, but actually the true one is here, all cis. We should write all cis, then 9, 12, 15. What means all cis? Because all are in cis operation. Assume the one is in trans. For example, 15, then we should write all cis will be incorrect. And the correct one is, for example, cis 9, cis 12, trans 15, octadecatrianoic acid. So one by one, we will write the configuration just in front of the number of position of the double bond. And also remember, for organic chemistry, we always use the carbon number which the double bonds begins, not ends. The naming of carbon atoms in fatty acids follows the Greek alphabetical order, so which means each carbon atom or some carbon atoms in the fatty acid chain have a name, have a Greek alphabetic name. And the carbon atom after the carboxyl group, which is the first group, it's called the alpha carbon. And the following one are beta, gamma, and then delta, and then goes on. At the farthest carbon atom, it doesn't matter how many carbon atoms are there in the structure. The farthest carbon atom is always the omega carbon atom, which is the carboxyl, which is the terminal methyl group. Methylate. There may be 10 carbon fatty acid, there's a 10 carbon fatty acid or 18 carbon acid, and in both carbon fatty acids, the terminal group is called as the omega carbon. The methyl carbon here is the omega carbon. And look at the palmitic acid here. And so it's very easy in skeletal structure. This is the carboxyl group, what I said. Before explaining, let me to finish. Yes, okay. Yeah, this is the first carbon atom, the carboxyl carbon. And this information comes from the organic chemistry courses also. The carboxyl group, the carbon atom of carboxyl group, or the carbon in carbon is the first carbon. And the other ones follows second carbon, third carbon, fourth carbon, fifth, until to the end. One by one, count the number of carbon atoms, and in palmitic acid, it ends with the 16 carbon atom, which makes the 16 carbon atom the omega carbon, and it is the methyl group. And its identity is 16 colon 0. As you see, there is no any double bond. And the beta carbon has a significance, especially in the metabolism of fatty acids that we will discuss in the Biochemistry-II. And omega is a special carbon because there is an omega classification of unsaturated, not the saturated ones. And to study nomenclature, you should remember the UPAC numerical multipliers. We have to remember. You know that we need to use these numerical multipliers when we specify elements like carbon chain, the number of carbon atoms in the carbon backbone, or also in functional groups and the substituents. And here, as you see, from 1 to 4, remember, as you see here, the mono D3 tetra, if you want to use numbering 1 to 4 as a substituent or as a number of functional groups, number of substituents, we use mono D3 and tetra namings as seen here. But if there is a carbon backbone carrying 1 or 2, 3 or 4 carbon atoms, instead of mono D3 tetra, we have to use MEF, F or at, prop, and but. So remember this difference. And especially till to pentacosa, you should know, especially in studying fatty acids. And the common ones are what I said, the octadeca ones and the hexadeca ones. But the aeco ones has a different or has a importance and additional importance because we have the eicosanoids. They are the derivatives of the fatty acids carrying 20 carbon atom. Numbering and display of unsaturated fatty acids can be done in two ways. The one is starting from the carboxyl head. So here, what I'm talking about is numbering or displaying. Or I say they are kind of identity numbers, giving the identity numbers which are unique. As remember, think about your identity card. Each of us has a special identity, a unique identity number belongs to us. And this is similar for fatty acids. But there are variabilities. There are two possibilities, an identity giving them according to the carboxylic head, or in other words, giving according to the UPAC numbering style, or systematic rules, following the systematic rules. And the second type is according to or starting from the omega carbon. And this is done for unsaturated ones. Be careful, unsaturated ones. So, starting from the carboxylic head, there was a double bond between the 9th and 10th carbon atoms in the majority of the unsaturated fatty acids. So, starting at carbon of carboxyl carbon, the carbonic carbon atoms are numbered. The carbon atom in which the unsaturated bond is located is designated delta sign. And the atomic number of the carbon atom, actually not the atomic number, the position number of the carbon atom, I will correct it later, to which the bond begins is written as delta 9. If it contains more than one double bond, the double atom at which the double bonds begin is written by adding a comma, as seen here, delta 9, 12, 15. It must be comma. If it is to be written without specifying delta, so this means there is an additional style. The style is first write the number of carbons using colon, then put the number of double bonds, and then put a semicolon, and write the positions of double bonds, separating them with comma, 9, 12. Let's look at an example. Here is a fatty acid given to us. And what is asking? Number and name the fatty acid starting from the carboxyl head. So, number from the carboxyl head. Name from the carboxyl head. And there is a question mark here and also an exclamation mark. I put the naming always follow the systematic rules. Always follow the carboxyl. There is no naming according to the omega carbon. The omega is just a classification. Be careful, my friends. So here, to name, to number, firstly identify the carboxyl. This is the carboxyl group, which is the first carbon, and follow the second, third, fourth, and when we count the number, we will find there was a double bond between 9 and 10 and 12 and 13, and at the end it is this 18th carbon atom, which is the omega carbon. So in the fatty acid there are 18 carbon atoms. There are 2 double bonds on the 9th and 12th carbon atoms. Don't forget, always give the position of carbon atom which the double bond begins. And there are 2 styles, which one is using the delta, represents double bonds as a notation over delta, 9, 12, and then write the number of carbon atoms in the chain, and using colon, number of double bonds. So this is identity. Or the other one is write the number of carbon atoms, putting colon, then number of double bonds, semicolon, and the position of double bonds, separating with comma. And in the given example, there are 2. If there are 3, again continue by using comma, write the position, and also make here 3. So the name, look at 18. What is a multiplier? Octadeca, and it's an enoic acid. Octadecanoic acid. Is it correct? No, because there are 2 double bonds. It should be octadecadienoic acid. And also the name must contain the position of double bonds, which make it 9-12. The position of double bonds, it must be always following the carboxyl head, not the omega. Octadecadienoic acid. So the enoic double bonds, there are 2 double bonds. Octadeca, 18 carbon atoms in the chain, and 9-12 the position. And also there is one missing point which omit here. We know they are in cis configuration, so also we should put here, we must actually, all cis 9-11 octadecadienoic acid. And the common name is a linoleic acid. This is a PUFA, and this is a essential fatty acid, linoleic acid. The second style is starting from the omega carbon atom, which is the methyl terminal end. Unsaturated fatty acids are sometimes classified as omega acids or N acids and represent the omega here. And carbon atoms are numbered starting from the omega carbon or N carbon. So there are 2 styles as in the carboxyl head. And the numbering is started from the omega carbon. This is numbering and this is classification, not naming. Naming always follow the UPAC rules, systematic rules. The carbon atom number of the first double bond indicates the omega group or indicates the omega class of fatty acids, such as if there is a double bond at the third carbon, when we start from the omega carbon, it is an omega-3 class of fatty acid. Then with the order, the total number of carbons using colon, followed by the number of double bonds, followed by semicolon, and the number of carbon atoms at which double bond starts separating with comma. And let's look at the example. And as you see, we have the same fatty acid, but here number and name the fatty acids from the methyl terminal end. The methyl terminal end is the omega. So the question tells us number and name according to the omega carbon. But what I said, be careful. The name is always given the UPAC. So this is a tricky question. Yes, number according to the omega, but still give the name according to the UPAC. There is no naming. Yes, at the end, the carbon chain name is same, octadeca-3-enoic acid. The difference occurs in the position of double bonds. In the UPAC name, the position of the double bonds must be given according to the following to the carboxyl head, not the omega. Be careful, my friends. Be careful. And yes, identify the methyl end, which is the first carbon according to the omega numbering, and count or number the carbons following 2, 3, and go on to find the first double bond 6 and the second double bond at the 9th position. As a total number is 18, the same. So there are 18 carbon atoms. According to the terminal end, the double bonds are on the 6th and 9th carbon atoms. And the first double bond, which is the 6th, determines the omega class of given fatty acid. This is omega-6 fatty acid or N6 fatty acid. So there are 2 styles. If we do and numbering according to the omega carbon, the numbering must start with the omega class. This tells us the given numbering, the positioning, done according to the omega carbon. Because now we have a formula and we put the numbering. And inversely, I can give you a numbering and then expect you or ask you to draw the molecule or to name the molecule. So you should know, you have to know which type of numbering is followed. And the differentiation is here. If there is no omega class in front of the total number of carbon atoms, we understand that the given numbering is done according to the carboxyl head, not the omega. If it is omega, the omega class must be assigned in numbering. And then as a separation, there is a carbon symbol, and the following style is same with the carboxyl head, but be careful, the positions are given according to the omega carbon. And a hint for you, if we have an omega-6, which we know this is the first double bond we see in the chain, so here also after the semicolon, the position of the first double bond must be same. So if it is 6, it must be start with 6. Assume there is a omega-6 is given to you, but here instead of 6 there is a 3. So this is a sign that there is a problem. Either one is incorrect. And the second style is, instead of using omega alphabetical letter, we can use N-6 using comma, also here comma, then number of carbon atoms, and be careful, if we use N style, we don't put the carbon C here, and the following is same. Colon, number of double bonds, semicolon, number of position, the position of carbon-carbon double bonds, following omega. And the epoch name is same, it doesn't change. 9-12, and here in the name 9-12 follows the carboxyl head, be careful. Octadecadenoic acid also is cis, all cis octadecadenoic acid. This is the famous linoleic acid. Again, I want to emphasize, remember that the epoch name is always given according to the carboxyl head. We mean the double bonds here in the name, the position of double bonds. So be careful, my friends. And my dear friends, here you see them all as a whole. Both numbering style, omega in 2 different style and carboxyl in 2 different style. And also in the structure, you can easily understand double bonds are in cis configuration. If they were in trans configuration, the style would be like this, in a kind of zigzag drawing, as you remember from organic chemistry. But here it is cis, and all cis. Let's take one more example, my friends, and here is another fatty acid, and now it will be more easy to study it. And as you see, there are 18 carbon atoms, and the blue ones are according to the carboxylic head, and the red ones are the omega carbon. And from the carboxyl head, as you see here, 1, 2, 3, 4, 5, 6 carbon, 9, 12, 15 carbon atoms carry double bond in the cis configuration, as you see, all cis. And from the omega, 3, 6, 9, and 12, and also there are 4 double bonds, which make it tetra anoic acid. And also the omega class is omega-3, because we have the first double bond at the third carbon atom. So according to the carboxyl head, the identity is delta 2 6, 9, 12, 15, and don't forget to put the commas, 18 carbon atoms, colon, and 4 double bonds, or 18 carbon atoms, colon, 4 showing the number of double bonds, and then with semicolon, we will put the position of double bonds according to carboxylic. And it's omega class, or according to the Fermilan end, and as you see there is a methyl group. It's omega-3 class, putting comma, then carbon, 18, 4, 3, 9, 3, 6, 9, 12. And don't forget the hint, if it's an omega-3 class, the first double bond must be at the third carbon atom. Or N3, another style, using comma, then number of carbon atoms, and as you see here, there is no C, the carbon symbol, and the rest is same with that style. So its name is, my friends, 6, 9, 12, 15, octadeca, representing number of carbon atoms in the backbone, 18, tetraenoic represents there are 4 double bonds, acid. And also don't forget, we have to put all Cs, because all are in cis configuration, 6, 9, 12, 15, octadeca tetraenoic acid, and its common name is stearidonic acid. And it's another polyunsaturated fatty acid, the PUFA, belonging to the omega-3 fatty acid family. And it is considered an intermediate in the biosynthesis of longer chain omega-3 fatty acids such as eicosapentaenoic acids, the EPA, which is timnodonic acid. And yes, another example, which is the EPA. So the previous one is an intermediate metabolite during synthesis of that one. And as you see here, there are 5 double bonds in cis configuration. All are in cis configuration. And this is omega-3 class. There are 20 carbon atoms, which make it aeco. And from the carboxylate, we have the double bonds, 5, 8, 11, 14, 17. And from the omega, we have the double bonds at the position 3, 6, 7, 8, 9, 10, 11, 12, 13, 14, and 15. And this is according to carboxylic head, delta 2, 5, 8, 11, 14, 17, 20 colon 5, or 20 colon 5, semicolon, 5, 8, 11, 14, and 17. According to the methyl terminal end, this means according to the omega carbon, omega-3 class, using comma, then the symbol carbon, 25, 3, 6, 9, 12, 15. And don't forget, if the class is free, the first double bond must be at the third carbon atom. And this is another style. And so the name is, again, I forget to put the all C's, 5, 8, 11, 14, 17, eicosapentaenoic acid. And this is the timnodonic acid, or shortly EPA, eicosapentaenoic acid. Another polyunsaturated fatty acid acts as a precursor in some eicosanoids, or as an intermediate metabolite during the synthesis of prostaglandins, thromboxane, etc. And the human body converts some of the absorbed alpha-linolenic acid into EPA. EPA is also precursor of docosahexaenoic acid, which is the cervonic acid. Yes, another example, and as you see here, we have a fatty acid carrying 4 double bonds, which make it tetraenoic acid. So this is the suffix, and then count the number of double bonds and give the identity according to the carboxylic head. Here's the carboxylic head. This is the methyl terminal, and then count 1, 2, 3, 4, 5 1, 2, 3, 4. Yes, this is 5. 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 12. So there are 12 carbon atoms, and this is the omega. And as you see, according to carboxylic acid, we have the 5th, 8th, 11th, and 14th positions. There are double bonds in cis configuration. And so delta to 5, 8, 11, 14, 24, or 20 colon 4 semicolon 5, comma 8, 11, 14. And according to the formula, and this is the omega carbon, and the class is omega 6. So at 9, 12, and 15, there are the double bonds. And this is omega 6 class of fatty acid carrying 20 carbon atom. And don't forget, if we use the letter omega, we have to put the carbon C and following colon 4 semicolon 6, 9, 12, 15, and this is another style, and the name is, again, don't forget to put the all C's. We have to mention the configuration of double bonds. 5, 8, 11, 14, eicosahexatetraenoic acid. And my dear friends, this is the famous arachidonic acid, and it belongs to the eicosanoid family. And the molecule synthesized from oxidized polyunsaturated fatty acids to mediate cell-to-cell communication. And eicosanoids contribute a lipid signaling complex that is commonly responsible for triggering an inflammatory immune response. And arachidonic acid is a polyunsaturated fatty acid found in phospholipids, especially in phosphatidyl-ethanolamine, phosphatidylcholine, and phosphatidyl inositones of the membranes of body cells, and is abundant in the brain, muscle, and liver. And skeletal muscle is a particularly active site of arachidonic acid retention. However, it becomes essential if there is a deficiency in linoleic acid or if an inability to convert linoleic to arachidonic acid. And some mammals lack the ability or have a very limited capacity to convert linoleic acid to arachidonic acid, making it an essential part of their diet. And we will talk about the essential fatty acids in the following slides. So, this is for you. Please study and try to number according both carboxylic and methylene, and also give the name of this fatty acid. And a hint for you, there are 20 carbon atoms. This is docosapentaenoic acid, the common name. And this time, let's draw a fatty acid called coulopanadonic acid, another polyunsaturated fatty acid. As you see, there are 5 double bonds and all are in cis position. And it's a docosapentaenoic acid, which means there are 20 carbon atoms in the backbone. It's penta, so 5. It fits with the given positions, number of given positions. There are 5 positions. And enoic acid, also referred, it's a carboxylic acid and carrying 5 double bonds. And this is the coulopanadonic acid. So don't forget to represent the all C's. The drawing style must be like this. If you draw like this, it means trans double bond, so it will be incorrect if the given molecule is all cis. Another example, there may be, for example, 7 in the cis configuration, 10 in the trans configuration. So to draw, you must draw the 7 as a cis configuration and following 10 in trans configuration. Classification of fatty acids. We can classify them as saturated fatty acids and unsaturated ones. And here you see the saturated ones and the stearic and palmitic, the common ones in nature. And acetic acid, propionic, and butyric, these are the common volatile fatty acids. And as well as still 10, that fatty acids are volatile in room temperature. And also liquid for the saturated ones. But the bigger ones, which are the abundant ones, they are solid. And the second class is unsaturated fatty acids, UVA, and we have the PUFAs, and we have the MUFAs, the polyunsaturated ones and the monounsaturated ones. And the common one in nature is the oleic acid, and except the allylic acid here, as you see, and also the waxanic acid. This is also trans. The rest are in cis configuration, and especially the PUFAs, which are the omega 6 and 3 ones. Here, 3, they are accepted as essential fatty acids. But in real, the linoleic and the alpha linolenic acid, these are the two real essential fatty acids. And also we have the omega classification. And there are four classes of omega 3, 6, 7, and 9. And remember, omega is for the unsaturated fatty acids only, unsaturated. And my dear friends, did you notice something? Look at the given fatty acids. Look at the left and right part of the slide. And yes, I hear that you realize the omega 3 and omega 6 class of fatty acids are all polyunsaturated fatty acids. And omega 7 and 9 class of fatty acids are all monounsaturated fatty acids. And as I said, the 3 and 6 referred as essential fatty acids. And also, according to the number of carbon atoms in the carbon chain, we can classify them as short chain ones, medium chain ones, the volatile ones, and especially these are the famous ones, which are important in the ruminant metabolism because both acetic, propionic, butyric, and as well as other ones in minority or during the synthesis of other fatty acids or breakdown of the bigger ones, because I said the long chain ones are the common ones. In ruminants, acetic, propionic, and butyric acid they are the end products of carbohydrate fermentation by symbiotic microbes in the rumen. And ruminants can utilize these fatty acids to yield energy, to synthesize fatty acids, and also to synthesize glucose. And here the propionic acid is the glucose precursor in gluconeogenesis that we will discuss in the biochemistry to gluconeogenesis. Yes, a topic for essential fatty acids, a title. They were first detected almost 70 years ago. The term essential fatty acids refers to polyunsaturated fatty acids, which must be supplied by food because they cannot be synthesized in the body but are essential for health. In other words, they are fatty acids that cannot be synthesized by the organism and must be taken from outside with food. In general, omega 3 and omega 6 unsaturated fatty acids are considered essential fatty acids. Special, however, and they are, and don't forget we talk about the mammals. However, among these, alpha linolenic acid and linoleic acid are the true essential fatty acids. And alpha linolenic acid, which is the omega 3 class of fatty acids, is also the precursor of the other series of omega 3. And linoleic acid, which is the omega 6 fatty acid, is the precursor for the rest of the omega 6 class of fatty acids. But there is a differences between species for sure. It always happen. And but in general, we accept, as I said, all are all 3 and 6 class of fatty acids are essential because depends on the diet content, depends on the metabolic processes, they may be insufficient. So they must be supplied by foods. Some disorders occur as a result of inadequate or no intake with food, and they are characterized by skin disorders, lesions, dry skin, and growth retardation. Essential fatty acids are necessary for the synthesis of eicosanoids, prostaglandins, thromboxanes, leukotrienes, and the other ones. And these compounds have hormone-like effects. Some also say they are hormones. They are present in trace amounts, but their physiological effects are very important. Yes, another title, Physical Properties of Fatty Acids. Both the physical and physiological properties of fatty acids depend on the length of the carbon chain and the number of double bonds in the molecule. In other words, the degree of unsaturation of the fatty acid. Fatty acids are amphipathic and have both hydrophobic and hydrophilic regions. This dual structure often plays a key role in the function of biological lipids against water. Hydrocarbon groups try to combine to form a minimum surface with the carboxyl groups forming a contact surface around the mass of water. The chain length of the hydrocarbon group is determined by superior behavior, and hydrophobic structures are very strong, especially in saturated ones, for example, palmitic acid. Fatty acids are not water soluble, but their sodium and potassium salts, which we call them soaps, are soluble in water. The sodium and potassium salts of fatty acids emulsify oils and water in soluble fatty substances. Sodium and potassium soaps dissolve in water and they are used as cleaners. But others are not soluble in water and are not cleaners. Commercial soaps are sodium salts of palmitic, stearic, and oleic acid. Calcium and magnesium soaps or fatty acids are very difficult to dissolve in water and thus cannot emulsify fatty layers. When potassium soaps are used in hot water containing calcium and magnesium, they do not dissolve in the water and precipitate to the bottom. All saturated fatty acids with carbon number up to 10 are liquid and volatile at room temperature. Fatty acids with carbon number higher than 10 are solid at body temperature. As the carbon chain of fatty acids increases, the fatty acid begins to harden and its melting point increases. Unsaturated fatty acids are liquid at room temperature due to double bonds in this structure, due to cis double bonds in this structure. In addition, as the number of double bond increases or number of cis double bond increases, the melting point begins to decrease. And here is a table showing some saturated and unsaturated fatty acids. actually the common ones and also the essential ones. These are the PUFAs, and this is palmitoleic and oleic acid are the monounsaturated fatty acids. And compare the number of double bonds. For example, look at the stearic acid and arachidic acid, and having the same number of carbon atoms. Look at the oleic, linoleic, linoleic, and the arachidonic acid. And especially these three ones having, despite being carrying the same number of carbon atoms, due to their number of double bonds, they have different melting points. And as you see, increasing the double bonds, look at the arachidonic acid, there are four, the melting point decreases, look at here, minus 49. What about arachidic acid? 77. Number of carbon atoms increases, melting point increases in saturated. In practice, natural acyl glycerols contain a mixture of fatty acids that are shaped to suit their functional roles. For example, membrane lipids, which should be liquid at all environmental temperatures, contain more unsaturated fatty acids than storage lipids. Lipids in tissue exposed to cold, for example, in animals living or hibernating at the poles or in the extremities of animals, are more unsaturated. In other words, living beings living in cold regions have more unsaturated fatty acids in membrane lipids than living in warm regions of the world. Glycerides, which are rich in saturated fatty acids, are solid, but the membrane lipids are rich in unsaturated fatty acids. Almost all of the naturally occurring long-chain unsaturated fatty acids are in cis configuration. However, in unsaturated fatty acids, although isomers are formed by changing the location of the double bond, the more common isomer form is the cis and trans isomers that arise depending on the arrangement around the double bond. And this affects the melting point. For example, the oleic acid has a melting point of plus 16 degrees Celsius, and we know it's a cis fatty acid. If we treat the oleic acid with nitric acid, it transforms occurs, which is the allylic acid, and look at the melting point is plus 45 degrees Celsius. And the last title, chemical properties of fatty acids. And fatty acid salts, salts made by fatty acids with carbon number higher than 6 with metals are called soaps. The ionized head parts of soaps form a polar group and make hydrogen bonds with water. The nonpolar tail parts are gathered together, thus soap molecules disperse in water form micelles. And I have already mentioned the physical properties of sodium and potassium soaps in the previous slides. And we know the others do not dissolve in water and are not cleansimatic. The sodium and potassium has cleaning feature. Potassium soaps are softer than sodium soaps and melt more quickly. And soaps made from unsaturated fatty acids dissolves more in water and alcohol than the saturated ones. And the potassium salts of palmitic, stearic, or oleic acid are known as Arabic salt. And soap salts in the market are sodium salts of the same fatty acid. They contain sodium carbonate and sodium silicate to remove water. Detergent formation. Detergents are metal, such as sodium, potassium, calcium, magnesium, salts of sulfuric acid, esters of high alcohols of fatty acids, which are the reduction products. Since detergents are composed of calcium and magnesium salts, they can be used more easily in hard water. Because in hard water, the cleaning ability is more, especially in the detergent form. The fatty acid in the detergent formation, the fatty acid is first converted into the alcohol, and due to the long carbon chain. We call it high alcohol. And this process takes place with the high pressure and the heat. And then they are esterified. Since they are esters of sulfuric acid, they are superior cleaning agents due to the fact their salts do not disintegrate in acid solution. And since they do not disintegrate even with acid, they cause accumulation in nature and thus environmental pollution. And since some of the detergents contain potassium, this type of detergent is of great importance due to its polluting effect on the environment. Because potassium is a good growth factor for the proliferation of the green algae, which they live in fresh waters. And with the mixing of detergents into the environmental waters, the reproduction of alkalis in the waters also increases, thus the amount of oxygen in the waters decreases. And the decrease in oxygen in the water will cause the death of aquatic organisms, especially fish, and so it will cause the pollution. Ester formation. Carboxyl groups of fatty acids are irreversibly combined with alcohols. Esterification occurs slowly, spontaneously, but rapidly in the presence of heat or hydrogen ions. Properties of double bond and hydrogenation. It is the saturation of the ethylene bond in the structure of unsaturated fatty acids with hydrogen. For example, if oleic acid is hydrogenated, stearic acid is formed. Thus, unsaturated fatty acid becomes saturated. The halogen addition to the double bond. Double bonds of unsaturated fatty acids are saturated by the addition of halogen. And this process takes place at high pressure in the presence of catalysts such as bromine, nickel, fluorine, chlorine, etc. Oxidation is the oxidation of the ethylene bond, and potassium permanganate is used as oxidant in the oxidation of fatty acids. Since the oxidation process is very complex, many intermediate metabolites may also occur during oxidation. If the oxidation agent is ozone, ozonate is formed by adding to double bonds. These reactions are used to determine which carbon atoms are joined to double bond in an unsaturated fatty acid. And here we are at the last slide. Peroxidation. Peroxidation of fatty acids, especially polyunsaturated fatty acids, which occurs in vivo and is called the lipid peroxidation, is a chain reaction. And during the reaction, very reactive free radicals containing unpaired electrons are formed. And free radicals enter into large-scale reactions in the living organism. However, the defense system in the organism prevents or controls the harmful effects of these compounds by antioxidative effects. So we can say the free radicals are highly toxic for the cell. And the peroxidation of lipids in the cell membrane causes disruption of the structure of membrane proteins or membrane lipids. And yes, my dear friends, the topic ends here. And these are the reference for you for further study. And the next topic is Lipids-II, part 2. And for more about biochemistry, clinical biochemistry, and lab world, you can visit my website. You can follow my Instagram account. And my dear friends, please do not forget to subscribe to the channel, like the videos, and turn on notifications to get the updates from the channel. So see you in our next topic. Goodbye and stay safe.